So yeah, anyway, thank you very much for inviting me to this exciting talk. And I also want to say sorry for Carl's past. Yeah, I think I met him in Kyoto long time ago. I don't remember when it was, but yeah, I remember him. And yeah, actually, at our age, we lose suddenly, unexpectedly, very good friends. Anyway, so today I want to talk about memory system, or actually physics approach to memory. So as yeah, actually I was trained as a physicist, and so my first career started from Hopfield network. But before uh, going that, probably many of you do not know me, so I decided to introduce briefly my laboratory. And actually, this photo was taken today. Yeah, and Okinawa is very fine. But you, this is, yeah, today is probably the first day we got to this kind of fine weather. Okinawa actually is fine only during the summertime, and other maybe 10 months or 10 months, weather is something like in England. Yeah, you may not believe. But anyway, so my uh, yeah, our lab's interest is computational mechanism of memory and other cognitive function. And so there are many reasons to study brain function, but so there are intriguing features: brain computing, energy efficiency, and data efficiency compared to AI. And brain circuit is networks of functionally complex neurons, not simple neurons. And brain uses spikes. And these days, my lab is interested in spontaneous dynamics of the brain. Yeah, brain never stops as far as we live. And spontaneous activity is not just a noise. And we, of course, brain is highly parallel and simultaneous sequential processing. Yeah, and all these features we still have to understand. And so we are combining theory and data analysis. Yeah, to understand these issues. And but today, yeah, I want to talk about kind of associative memory related topics. There are few reasons. One is, as you know, Hopkins got the Nobel Prize, not in biology, but in physics. And I was happy because my career started with Hopfield network. And another reason is this model is now people know that relationship between Hopfield kind of associative memory and large language model. Yeah. So interests are reviving. And so I basically talk about two topics. Yeah. One is so graphically, so how you can embed relationship in associative memory. Actually, memory items are usually not isolated. I mean, there are relationships, and but how these things relationships are embedded are not well known. And second topics is so non-attractor associative memory. And this I will explain you later what's the concept. But anyway, in both of these topics, inhibitory circuits play crucial role. By the way, can you hear my voice clearly? No echo? Is it okay? Yeah. So the first topics. So basically, so actually, this map is the town I'm living. So I'm in Okinawa, small island. The Okinawa is like this, and I'm living in city called Okinawa City, and this is a map of Okinawa City. And actually, I'm somewhere here, and this is not my room, but usually my room is separated into several pieces. And so memories, all these memories of room or town are connected, and yeah, in a hierarchical fashion. And so I want to know how this can be possible. Of course, there are a lot of models for hierarchical memory, but what I want to, before that, I don't know, maybe some of you may not know what is associative model. So I just give very brief explanation. Yeah. So if you know, please sleep. So basically, Hopfield network is the current network, and there are connection matrix called W, and this is usually symmetric. And you introduce binary neurons, so this can take either 1 or 0. And what network is doing, or each neuron is doing, is sum up input, synaptic input, weighted synaptic input, and make a thresholding operation. And then, next step will take 1 or 0. And what Hopfield suggested, or actually showed, was you can define so-called energy landscape. And so this is always minimized. Yeah. So change is always negative. So if and so finally the state will be settled in some attractor states. So this was basic idea of attractor network. And in my opinion, there are no direct still evidence for attractor dynamics, although there are many claims. But in any case, this idea actually give a big impact even in neuroscience. Yeah, and so as I said, what this model suggests is you can actually embed some number of finite number of memory patterns, and this can be so binary pattern again, and these memory patterns can be made local minimal of energy landscape if you, for instance, define connection matrix between neurons. Yeah, by doing this. Yeah. So this is called local hip model because it is depending on the activity of both postsynaptic neuron and presynaptic neuron. So some analogy to so-called hip rule. And there are a lot of bunch of study about this model. And so this is a very recent one. So basically, by downhill motion, you can retrieve embedded pattern, one embedded pattern. But there are a lot of, if there are similar pattern, then sometimes error occurs, and something like that. And physics show that the capacity of your network has finite memory capacity. You cannot embed infinitely many neurons. Many patterns. And these days, Hopfield himself proposed generalized Hopfield model and showed the relationship to large language model. Okay, anyway, but what we are interested in is actually relationship, memory of relationship, and actually there is some Q already in 1988. Yeah. So what this person showed is he showed these images into monkey and recorded the activity of visual cortex, some visual, near IT cortex, inferotemporal cortex. And then he found each stimulus causes some, induces some kind of activity pattern. Okay. And of course, activity pattern are different for different stimuli. But interestingly, these activity patterns are mutually correlated. And so correlation looks like this. So this distance of two stimuli along the sequence. So this is you can define the sequence of a presentation, right? Then what they suggested, showed is actually correlation or overlap between attractor drops gradually and disappear if the distance is more than 5. Yeah, and so this attracted actually, group of Daniel Amit, and they proposed a beautiful model using attractor network. So what they did is actually, so this is the original Hopfield network now, okay? But they introduced some kind of flow term. So this is a flow from pattern mu to mu plus 1, and this is opposite flow. And minimum requirement of to have energy, downfield motion or energy landscape is, so W should be symmetric. So this actually these new terms keep synaptic potential symmetric. Okay, and actually they showed this model replicates this feature. So if pattern distance between pattern becomes more than 5, then correlation disappears. Yeah, and that kind of, so when auto association is stronger, then Hopfield type uncorrelated attract appear. But if this term becomes weaker, then new correlated state appear. Okay. And one thing we notice is, so because we are considering sparse activity pattern, you have to subtract some bias. So this is a probability that this pattern takes place at 0 or plus 1. Yeah. Then, this is in some sense adjusting a bias in the pattern. Therefore, it is interpreted as EI balance. Yeah. Okay. Then what we noticed, so actually this is one of the very beautiful example where two models, associative memory model, actually nicely explain the results. But so recently, so this is fear conditioning, and so that learns two contexts. So this is some arena, and so in one arena, shock is given. But in that kind of situation, so if you observe activity of CA1, hippocampal CA1, then you find many neurons are activated. But actually overlap between neuron ensemble, activated neuron ensemble decreases with time. So if two contexts are shown 5-hour difference, then overlap, you have to observe relatively large overlap. But if this is 7-day, overlap disappear. So actually, it seems temporal correlation, temporal relationship is somehow embedded into spatial overlap. Okay, and what we showed is actually previous so Amit's group, they only consider the range where order association term is positive. But because already this can support memory, so in principle, we can make this coefficient negative. And actually, if something interesting happens, if this you allow auto association term, so this is this connection is within pattern. So neurons within pattern are connected, and if this becomes negative, actually distance becomes much longer. So when So this case is previous case. So distance is just 5. But if C becomes negative, actually distance becomes much longer. And this example is 15, but 15 is not the maximum. You can make it much longer. Yeah. So we found this several years ago. When I joined OIST, and we called it extended correlated attractor. Yeah. And if auto association term becomes too negative, too weak, then there is no memory states. By the way, so this is auto association term, and we call second term, flow term, heteroassociation term, because different patterns are correlated. And then we further analyzed, and we found actually this has some different interpretation. So as I said, we subtracted average activity level P from each pattern, and this can be interpreted as EI balance. And so we explicitly write this into weight matrix by the sum of excitatory connection and inhibitory connection. Then excitatory connection has no interesting feature. However, inhibitory connection actually contains important parameter C. So the first term, actually, this term depends on the pattern within each, so or activity of, let's say, cell group mu pattern mu. Okay. So this, in some sense, this is can be regarded as engram specific inhibition. On the other hand, second term, this is just sum of all assembly, activity of all assemblies. And so this is engram non-specific. Okay. And the balance between these two inhibitory terms are changed by C. So if C becomes -2, so this is a lower bound, then this term disappears. So the inhibition becomes engram specific dominant. On the other hand, if C becomes 0, then these two terms are included with equal weight. So this is C equals 0 corresponds to balanced engram specific and engram non-specific inhibition. Yeah. So, but changing C implies we are basically changing two types of inhibition. And if we go to engram specific inhibition, then correlation becomes wrong. So there are two types of inhibition, and engram specific, if engram specific inhibition is stronger than global inhibition, then many assemblies can be activated. So correlated attractor states contain many assemblies, while global inhibition or engram non-specific one is dominated, then only one correlation distance becomes 5. Yeah. So we consider that this balance between two types of inhibition actually controls the temporal range of association. Yeah. Okay. And we can further extend this model. And so for instance, this is my loop. Maybe I can embed this spatial environment by neurons. So here, each location of this room is embedded by some activity pattern, random memory pattern. And the relationship between spatial point is represented by this connection matrix. So this is auto association term, but the second term describes the relationship. So if this and this are connected, so A mu is 1. And this point and this point in the different room is connected, so A is again 1. But otherwise, if 2 points are unconnected, not connected, then this is 0. So it's called an adjacent matrix. Adjacency matrix. And as we showed, so this auto-association term is positive. So if animal is here, then this memory of this point space is retrieved. But if C becomes negative, then even if animal is somewhere here, this system retrieve information about all entire space point in this room and some in next room. And if C is further negative, then actually you can retrieve information about next room, even if you are not there. Yeah. So like this, by changing the balance between two types of inhibition, you can retrieve hierarchical or graph structure related memory. Yeah. Okay. And so this is just interim summary. So we defined generalized correlated attractor model. Yeah, basically this model. And this store graphically related memories, store and retrieve. Okay. And for the engram specific and versus engram non-specific inhibition balance regulates the range of retrieve episode. Okay. Yeah. Okay. I don't know. Actually, I have no watch. Yeah, anyway, I hope. Yeah, it's fine. So actually, I'm also So memory actually essential feature of memory is a relationship, and this is our most some of recent paper. So I think many of you know that the hippocampus is can be used important for spatial navigation. Yeah, place cell and and there is some years ago. Some people propose that actually the hippocampus is predicting the animal's movement. Yeah. So animal moves while it navigate this space, then it will predict where he can reach at the next moment. And so some people started to discuss about the relationship between hippocampus and reinforcement learning, which is proposed to describe the spatial ganglia or information processing of reward information. And basically, in this framework, we consider so-called temporal difference error or value of some state S, let's say some spatial point S, and it is sum of reward at this point, and then discount sum of future reward. Okay, this is the standard model, and about 20 years ago, this was a little bit more sophisticated theory was proposed that was called the successor representation. Yeah, so it is basically so the idea is just try to separate reward information and spatial structure. Yeah, the weak point of the original model is if reward position is changed, but even if state space, spatial structure of environment doesn't change, still animal has to learn everything from the beginning. However, that's very economic and not economic. So what Peter Dan considered is you define expected future state occupancy. So probability that you occupy state of S while you start from state S prime. And probability also discounted like this. Then you can show actually value of state S is product of state successor representation and the reward at the state S, and you just sum up this for all possible initial state. Okay, that leads to S. And so this is interesting because it tells, it allows you to describe relationship between state space, different state space. So this can be used for prediction. And what we did is, we defined successor information. Yeah, because successor representation is probability. Therefore, you can define information, then and try to find out optimal representation for this state space. Well, basically, neural representation. If you have n neuron, we consider pattern of n neuron to represent successor representation. And for doing that, so this is the first term. So representation vector for two states should be sufficiently good. So this should be minimized. And then, but the correlation between vectors should be also small. And also, we consider activity itself, activity level of representation vector also should be slow. Yeah, so we try to find out sparse representation of successor information. Okay? And of course, activity should be positive. The interest. So what we did is we first applied this to spatial environment. Then we could replicate so-called grid cells. Probably you know that the entorhinal cortex, neuron represents space. So one entorhinal cortex neuron responded to grid pattern, in the space. And you can obtain various type of grid representation, by minimizing the same, successor information. Okay. Then what we did is we applied the same framework to Wikipedia dataset. So now input is language sentences. Okay. Then again, representation is minimized. So now system try to find out relationship between the words. Yeah, if two words appears in relatively, in a close spatial point, then the system considers these states are related. And what happened is, so actually, for instance, unit number 31, this responds strongly to orbit spacecraft Saturn Apollo. So this is a space cell. Yeah. Then maybe, unit 244 COVID virus infected pandemic. Actually, we did this study during COVID. Therefore, Wikipedia full of COVID article. And we found virus cell. And actually, this kind of, so this is a response to name of the US president. So concept cells actually found in both hippocampus and entorhinal cortex a long time ago. And so we found our system can replicate both grid pattern and concept cells. Okay. And so, and this actually allows some kind of language processing, but I do not want to go into detail. So like this, anyway, we can, find the way to represent relations between memory items. Okay. So from now, I want to shift to other second topics. Actually, second topics and first topics are, I believe, related, but I will explain you the relationship at the end. So here, so far I discussed about four-field network. So the basic idea was, so there is attractor states, okay, minimum of the energy landscape, and network retrieves one of these attractor states. Okay, And but basically, I doubt whether that is actually the biologically plausible view. And so I explained why I thought things like this. So this is a typical belief that how hippocampus and neocortex work during memory consolidation, okay, or memory encoding and consolidation. So when animal is awake, so there are a lot of sensory input. So hippocampus probably store this information in the network. Then during sleep, then no sensory input comes. Then hippocampus actually shows spontaneous activity, or it is called replay activity. So the learned activity patterns are replayed spontaneously. And we believe this information is transferred into neocortex to generate kind of permanent memory. And during wakefulness, maybe we can retrieve this information from the engram or assembly stored in neocortex. So in this, if this is really the case, actually replay is important, but replay is not attractor state. So I'll show you. I don't want to go into detail, but this is my collaboration with some experimentalists. Yeah. So we studied activity pattern of CA1, unlikely CL, when animal is exploring some spatial, yeah, environment, and or during sleep after the exploration, and during retrieval of the experienced context. And we found in all stages of this task stage, actually activity patterns learned in this box are replayed. Yeah. Of course, other patterns, new patterns are replayed, but basically hippocampus is replayed many ensemble or population patterns, and they are not doesn't look stable attractor states. Yeah. Yeah, patterns keep changing. So let's consider then. So we do not ask. Yeah, again we consider recurrent neural network model of spiking neuron, but we do not ask attractor states, existence of attractor states. Okay, but we still embed memory activity pattern into connection matrix, just as in Hopfield network. But one big difference is now E2E connection is obeys log normal distribution. Yeah, there is experimental evidence that the synaptic weights are EPSP in CA3 and CA1, or even neocortex, obey log normal distribution, not the Gaussian distribution like Hopfield networks. So, log normal network connection has, I forgot to put the figure here, but unlike Gaussian distribution, there is a long tail. So log normal connection distribution allows very, very strong synaptic connection. Okay. Yeah. And but E2E connections are fixed. But here we introduce some activity-dependent plasticity into I2 inhibitory to excitatory connection to keep EI balance. We found this connection type is very important to keep stability. Okay? And we consider two types of learning rule. One we call it local balance rule, another we call it global balance rule. Yeah. So local balance rule actually each neuron is separately. So EI balance is imposed each neuron separately according to this rule. Yeah. Yeah. So inhibition is strengthened if yeah activity of pre and post activity is strong, correlated. On the other hand, global balance rule is you just so if n activity of entire network is high, then try to induce stronger inhibition. So that always, so global balance of the whole network activity is kept. Yeah. Okay. So details of this rule is not very important, but we are interested in the difference between local rule or neuron specific rule and network neuron non-specific rule. Yeah. Okay. And what we found is, first of all, so regardless of plasticity rule, inhibitory plasticity rule, actually embedded assemblies are spontaneously replayed. Yeah. So this is one example. So here we embedded, I forgot, maybe 1,000 patterns or something. Yeah. And I forgot to tell you. Yeah, there are 8,000 excitatory spiking neurons, and each assembly or each memory uses 80 neurons, and 2,000 inhibited spiking neurons. Okay. Yeah. And so this is 5 most frequently replayed assembly. Yeah. So you can see assembly. Yeah. So this is probably easier to see. Yeah. So this green one is reached almost unity. It means all neurons encoding one pattern, green pattern, is activated. But here, red one maybe 80%. So like this, yeah, patterns are replayed, and sometimes 2 patterns are replayed in overlap sense. Yeah. And another interesting feature is actually, so we tested whether if I stimulate, let's say 10% of pattern encoding neuron, whether pattern completion occurs during stimulus. And actually, it occurs. However, the quality of retrieval is much better if I consider local EI balance. Yeah. So local EI balance means each neuron, each target excited neuron, each postsynaptic excited neuron has target finding rate which is 3 Hz. Yeah. So inhibition becomes stronger if activity of this neuron, excited neuron, becomes higher than 3 Hz. And inhibition becomes lower if opposite happens. Okay. So yeah, these neuron specific EI inhibitive plasticity enables much better pattern completion. Yeah, but pattern completion occurs during when only stimulus is given, because there are no attractor states. So if stimulus disappear, then network returns to spontaneous active state. Yeah. Okay. But even without the stimulus, network spontaneously deactivate, replay one embedded pattern. Several embedded patterns. Okay. And another interesting feature was actually local EI balance is more unique assemblies replay then and at a higher overall replay frequency. Yeah. So here we change number of embedded patterns. Okay. And so each pattern I think is I forgot how many. Yeah. Anyway. So and so if number of pattern embedded pattern is increased, then number of replayed pattern also is decreased. However, for local balance, EI balance, number of replayed pattern is much higher compared to global balance. Okay. Yeah. Then similarly, so here I changed the number of embedded patterns, and we measured number of uniquely replayed pattern. And again, number of pattern is much higher for local balance rule, But of course, if number of patterns increase, then the number decreases. But this decrease actually doesn't occur very sharply. In attractor memory model, Hopfield network model, typically if you embed the number more than there is a critical storage capacity, and beyond that, suddenly retrieved states disappear. However, because there is no attractor states in this kind of network, that kind of phase transition does seem to occur very sharply. Okay. Yeah. And so we studied, actually this is still ongoing work. Yeah, we still have to complete the analysis. But because we made some analysis. Yeah. So we studied why global rule was the neuron specific learning rule. So for doing that, we introduced linearized, so we linearized the network model and studied the connection matrix, eigenvalue spectrum of the connection matrix. And random matrix theory tells eigenvalue should be within some circle. But and here, this red point, orange point is eigenvalues of global rule. And blue point is eigenvalue distribution of neuron specific rule, so beta rule. And when pattern is small, number of patterns is small, both looks nicely. By the way, there is an outlier here. So here there is no interesting activity in the network. So these three panels actually magnified most interesting distributions, this part. Okay. But if number is increased, then you see that some eigenvalue, positive big eigenvalue becomes greater or even beyond unity. And beyond this, actually, its activity becomes unstable. Yeah. And but you can see that this occurs only for orange point. So blue point is still within within the circle. And for pattern is 10,000, even blue one started to be disturbed, but compared to orange one, it's still nicely kept within the circle. So it means stability is strongly kept. So what this shows is, so spiking networks formulated for spontaneous memory replay. Yeah, so we made a spiking network model for associative memory. And for that, we only asked memory is replayed, but we didn't ask the presence of attractor states, stable attractor. And we found more assemblies are replayed when inhibitory plasticity is tailored assembly specifically or neuron specifically. Yeah. Okay. Actually, this is almost the last slide, and I don't know how much I spent. Yeah. But so I talked about generalized correlated attractor model. And also second topic, spiking non-attractor associative memory model. And we are now asking how these two may be combined actually. And what this generalized correlated attractor model is, so episodes in a closer temporal vicinity. Yeah, or episodes that occur closely in the temporal sequence, or then they are encoded by more overlapping attractors. Yeah, attractors are more overlap. Correlation between attractors are much higher. On the other, and then
spiking non-attractor model suggested overlapping cell assemblies are frequently replayed, meaning these patterns, if two cell assemblies are overlapped, then they are often simultaneously replayed. Yeah. Then, but this point, honestly speaking, we still have to prove more precisely. But numerical simulations suggest this. Then, if we combine these two, then what we can speculate is so temporally closer episodes are more frequently co-replayed. And because of simultaneous replay, then these temporally closer episodes are mutually associated during memory consolidation. And therefore, kind of events that occur within one episode might be nicely packed by overlapping cell assembly. And our model, both models suggest actually inhibitory microcircuit or inhibitory plasticity rule is important, crucial for regulating these processes. Okay, yeah, this is the last slide. And I finished my talk.